Hi, my name is Basil Al-Sheikhtib, and I'm going to present a framework known as FAIRClinical Structuring Clinical Report into OMOP Common Data Model. This framework is a part of a collaborative project among different academic institutions and universities, including the University of Luxembourg, the Swiss Institute of Bioinformatics, University of Leicester, and University of Paris-Saclay. This project aims at enriching the content of supplementary data and therefore enhancing the interoperability, findability, and reusability of such data. Basically, the area of parsing and analyzing biomedical literature has caught various researcher attentions in the last decade. However, little attention has been paid to the supplementary data associated with such publications. The challenge of handling such supplementary data is that it is highly heterogeneous, where different data formats can be found, including PDF, Excel, CSV, Word document, PowerPoint, and even images. In fact, there is a potential lies behind analyzing such supplementary data, where it may contain important information such as the Clinical Case Report Form, CRF. CRF is a detailed report of the diagnosis, treatment, signs, symptom, and the follow-up of a single patient. It is used to standardize the collection of these patient data in clinical research studies and the trials. The content of CRF are usually semi-structured, where a combination of structured categories of patient data along with free text can be found. We propose to process those unstructured CRFs to extract clinical concepts required to populate reference standard into structured common data model such as OMOP, which will directly improve the reuse of such CRFs. There are four main objectives which are stated as follows. The first objective is collecting and standardizing the publication with supplementary files, whereas the second objective is to identify the named entities within such CRFs. The third objective is to extract potential relationships among these entities and to structure the output into OMOP format. And finally, the fourth objective is to indexing and updating the enriched context. If we take a look at the first objective as shown in the figure 1, the first objective aims at building the CRF corpora in which the collection of publication can be used. First of all, we have to identify the search query, and for that purpose, we have chosen the genome-wide associated study as a starter query. And then upon such a query, the collection of the publication will take a place along with their associated supplementary files. And then there would be two separate filtration process. The first process of filtration aims at identifying the publications that have associated supplementary data, and this would be followed by the second filtration process in which the publication with supplementary files will be filtered in order to identify the ones that contain the CRFs. After that, a standardization process will take place in which the CRFs, the unstructured CRFs, will transform into a textual format. This would require image caption to text or table to text methods to be applied. For the second objective, and as shown in figure 2, supplementary files will start to be processed in order to identify named entities. For that purpose, we will leverage the state-of-the-art pre-training neural language models in order to do multidimensional named entity recognition. The first dimension contains administrative metadata, including author names, affiliations, licensing model, whereas the second dimension contains descriptive metadata, and here the biomedical named entities are going to be extracted, including diseases, gene or gene product, size of populations, and also other information regarding the experimental setting. The third dimension would contain structural metadata, including textual content and images. And finally, the fourth dimension would contain cross-references to other data repository or deposition catalogs. For example, here the extraction would take place for URLs or PIDs. For the third objective, and as shown in Figure 3, we will again leverage the state-of-the-art pre-training neural language models in order to do a relation extraction task. And here the aim is to identify potential relationships among the entities, more specifically the biomedical entities that have been extracted in the previous objective will now be undergo a processing of relation extraction in order to identify potential relations, biomedical relations. Specifically here, this may include protein-protein interaction or even gene diffusion. Such task will help transforming the unstructured CRF corpora into a common data model where the automatic population of the OMOP will take place. Finally, the fourth objective will provide an access to the enriched supplementary data collection through the SIB literature service. This can be depicted through updating the annotations already existing in CBILS with the new enriched content produced by the previous objectives. In the meantime, also indexing will take place in order to facilitate the search for the enriched structure CRFs. As a final conclusion, providing semantic enriched content is a key success for any potential dissemination or reuse of such content. This will enhance also the automatic retrieval and optimize the search itself by providing much more metadata and annotation. On the other hand, this would also open the door for relevant linking to external resources in which the content user would make sense of such content through the search, retrieval, and linking capabilities. This will significantly contribute toward content exchange, finding relevant information, and facilitating the accessibility of this information, leading to valuable contribution through creative reuses. Thank you.